Great. Hey, everybody. Good afternoon. So once again, thank you so much for joining us today here with at the CBTN. And so my name is David Higgins, and I'm the one of the program managers that we have here at CBTN involved in user training and outreach. And so today we've been put together this session to talk about different CBTN resources and tools and how they can specifically support early career investigators and researchers. And so we're all about supporting early career researchers. And so one of the first things that I wanted to ask everybody about is to get a little sense of a poll, is to learn about where are you in your career. And so we know that early stage can mean a couple different things, whether you're still a student or whether you are already have a position. Or you work in a, not necessarily on the tenure track, but still complete research as well. Just trying to get a sense of where everybody's at as we begin our session here today. So a few more ground rules before we get started is that our session's being recorded, and we're going to post this on our YouTube channel before we once we wrap up later today. But we also encourage everybody to participate. And so it's, I know that with the virtual medium, it can sometimes be hard to have that rich dialogue, but we do have our chat and Q&A feature, and we'll hopefully have some time at the end to get an answers as well. So please participate in the chat throughout the presentation, and we'll be able to answer your questions as we go, or hopefully at the end as well. And so our agenda today, so this is my quick introduction. And so then we're going to hear a little bit more about CBTN from Jenna Lilly. And then I'm going to be talking about our different resources that CBTN offers, including our biospecimen resources, as well as our data and tool resources. And then kind of interwoven between those, we've got two guest speakers, two users and researchers who've used CBTN data and biospecimens, Dr. Jess Foster and Dr. Adam Walker. And so those are our two different case studies. And then at the end, we'll have some final information about how to access CBTN data, as well as opportunities to collaborate in the future. So that's what we have to look forward to today. And so, like I said, we've got a full agenda. I do want to share the results of our poll. So thanks everybody for participating. Let's see, I don't know if it will let me drag it over to the other screen there. But we have some results. It looks like most of our attendees today are fellows, either postdoc fellows or medical fellows, as well as a smaller number of tenure track faculty, and then we've got 2 students as well. So thanks so much for joining everybody. And so that's a sense of who we've got in the room. So it's great to meet you all and learn more about your research needs. So at this point, I'm going to hand it off to Jenna. And so Jenna's going to tell us a bit more about CBTN and give us a great overview of what CBTN is and what types of advantages CBTN offers as far as supporting researchers, especially early career researchers. So take it away, Jenna.
Sure, thank you so much, David. And hopefully I'll be brief so we can get to the good stuff. But thank you all for being here today. We really look forward to supporting your work. Our challenge in the Children's Brain Tumor Network that we're trying to really spearhead and hopefully make change is that brain tumors are the leading cause of disease-related death in the US and also numerous countries around the world, but there's never been a drug developed to treat childhood brain tumors. So the Children's Brain Tumor Network was launched in 2012 to really spearhead this initiative. Next slide. We have a global vision. We are a research study, so we're an IRB approved protocol. We enroll subjects. We're a group of researchers, clinicians, patients, and foundations that has really come together to dedicate our efforts so no child dies or suffers from a brain tumor. Within the CBTN, we really have a policy of open science, global inclusion, and pioneering spirit because of the challenge that we pointed to earlier. Our data is available without embargo in real time. We want to make sure that all are available to participate. across the globe. We do have CBTN member institutions in Australia and in Europe and in China. And the pioneering spirit is for us to do this differently. We need to accelerate discoveries for children. And so our goal here in CBTN is to really support your research. Next slide. Here's a little snapshot of some of our member institutions. We started with 4 institutions. We're now up to 26. You can see our spread across the globe, and we're growing every day. So it's exciting to see new folks join. Member institutions supply biospecimens and longitudinal clinical data and subjects as enrollment in the CBTN protocol, and that is all shipped to the Children's Hospital of Philadelphia Operations Center. And so here at CHOP, we have the biorepository, the data repository, and the management of the protocol. And so for CBTN, we support researchers across the landscape, but we really do focus on this challenge of there's no biospecimens or preclinical models available. The CBTN collects, as I said, longitudinally from the day of diagnosis, biospecimens and clinical data throughout the patient's journey. We develop preclinical models, and David's going to tell us more about that. We also do foundational genomic characterization with whole genome, RNA-seq, proteomics, and some single cell. We also create those collaborative platforms for you all to come on and be able to compute across the genomics and share and collaborate. And then lastly, we do try to support each one of you. We know that you have a lot going on and you need to put in your grants and you need to do your work. And so we're here to support you to get you to the data or the specimens that you need and maximize any collaborations across the researcher network that we have with philanthropic entities and with federal grant opportunities. So with that, hopefully you can get a little snapshot here. We are the largest open access pediatric brain tumor resource. We've enrolled over 4,235 patients, and that's across all histologies. Publications David may go into. The publications are about over 150, and the publications are the researchers. When you use resources from a CBTN, you are the author list. We only ask for an acknowledgement, but hopefully this is your hypothesis and your research. We also enroll mothers and fathers, and so the total does increase to over 5,000 when you include those. Right now, we have 1,000 participants genomically characterized with whole genome and RNA-seq, and we just received a large NIH grant that is going to sequence the rest and the remaining of the subjects enrolled. So that will increase to another 4,594 whole genomes and 1,500 RNA-seq. We have now about 100 preclinical models, and we've just ventured into organoids, and those are available for your use. We do have 50,000 biospecimens. We've supported over 252 scientific projects across the world. And right now, as I said, we have about 26 member institutions. And I think David will go into more of those specifics, but all of these resources and are available to you. This is just a quick snapshot, and I think Dr. Foster might touch on this, but as you know, research takes a long time, and our goal here is really to accelerate discoveries for children today. So by providing the biospecimens, data, and support and infrastructure, our goal is to shorten the timeline for clinical impact. And you can see on this slide that from developing a hypothesis to actually enrolling patients on a clinical trial, we've tried to shorten that from the average of 12 years down to 4. And so we're hoping with that, we can get many more people interested in pediatric brain tumor research. We can get more research ongoing and some fantastic pharmaceutical partnerships as well, while getting these kids some new treatment.
Great. Thanks so much, Jenna. Yeah. And so one thing I really wanted to highlight about this slide, and this is my perfect transition into the next, is this collect sample data line. And so because the nature of the work of pediatric cancer research, these are very rare diseases. These are very rare tumor types. And so it can take a very long time to accumulate enough specimens from participant donors who choose to donate their brain tumor tissue in order to be able to further a research project. And so this is a real strength of CBTN, is that we have accumulated this collection across our different partner sites. And thanks to the generosity of our different participants who've enrolled in CBTN, that this is what allows us to be able to share this rich resource and give it back to the research community to go forth and do that great discovery that we so desperately need to try to make more progress against pediatric brain tumors. And so this is the first set of resources I want to talk about, is these biospecimen resources. And so, of course, I'm talking to a group of pediatric cancer researchers here. And so I'm sure you know that brain tumors are very, very complicated. They are one, of course, many, many different types of brain tumors. Within those many types, there are subtypes. And then even within the tumor themselves, there's different sections of the tumor that have different biology, different mutations, things like that. And of course, that brain tumor tissue is very precious, but also very depletable. And so by collecting this tissue, we can support researchers like you in order to be able to do that work that we need to be able to make that progress by having this significant number of tissues that's available for folks to be able to analyze. And so CBTN supports many different histologies. We enroll children and young adults in our IRB protocol with any brain tumor diagnosis. And so we are not limited to one particular type. And so we enroll participants, with some of the most common brain tumor types, like, for example, low-grade glioma, medulloblastoma, and high-grade glioma, which are among our have the most number of participants enrolled, but we also have much rarer brain tumors as well. So, for example, we have schwannoma, germinoma, and oligodendroglioma as well, which are the more rare. We have fewer participants, but we're still able to support research projects for these rare tumor types. And so, in all, we have more than 4,200 participants across all these types. And so if you study a single particular brain tumor type, you could read about it on our website, cbtn.org/histologies. And that's a great way to see all the different participants and projects we've supported based on different histologies. So when I say tissue, what types of tissue is actually available? And so in our repository, most recipients are asked for and receive frozen tissues. This is tumor tissue that's been taken out through an operation or during a procedure, and then is flash frozen and then preserved in that frozen solid state. CBTN also then uses that frozen tissue to try to generate cell lines, to try to generate PDX models, to try to generate organoids, and basically try to find ways that we can stretch that frozen tissue even longer. So of course, the frozen tissue is a limited finite resource, but if we can get a cell line going that we can proliferate indefinitely, then that would extend the value of that resource even greater. So we're constantly trying to take the tissue that we have and transform it into these more sustainable systems as well. We have FFPE, embedded tissue. We also have some slides that have been prepared from FFPE if you're interested in doing any immunochemistry. We also have fluids, so we have CSF and cyst fluid. And then we have germline tissue as well. So during an operation, we might collect a blood sample from a participant in order to get germline information. We also have parent saliva contributions as well. So that way we can look at parents and look at trio-based studies in order to be able to identify how perhaps different variants are being inherited, or the generation of new de novo traits in the patient themselves might be resulting in this cancer risk growth. And so how could we possibly use this? And so again, we're handing it off to you, our researchers, and so we hope that you can come up with ways, but ways that we've already supported it is that we have this large set of tumor tissue that can be used for drug discovery. So we've supported projects where they have collect a set of CBTN tissue and test their drug against those different cell lines in order to find growth, as well as different other treatments in order to be able to investigate how these different treatments might be able to affect our tumor samples. So ultimately, lots of different ways that we can support research. And so far we've supported more than 80 different scientific projects, all based on the biospecimens alone. And these are also on our website, cbtn.org/projects. So you can begin to see what other researchers have been doing with our data sets. And so we've reached the point now where many of these projects that were started a while ago have now reached the publication stage. And so we're excited to share that researchers are using CBTN data sets in these high-tier journals like Cancer Cell and AACR in order to be able to not just doing this discovery that can then be shared and published with the scientific community, which is outstanding. And so one of our members who is part of our scientific community is Dr. Jess Foster. And so I'm going to pass the mic to her a little bit and let her tell a little bit more about how she uses CBTN and how the biospecimens have benefited her work. Hi, Dr. Foster.
Hi, can you hear me?
Yes.
Great. Great. So yeah, so I can introduce myself. So I am an instructor in the Division of Oncology at the Children's Hospital of Philadelphia. I am a physician scientist, so I spend about 80% of my time doing research and 20% taking care of pediatric brain tumor patients. And my research is largely focused on CAR T cells for pediatric brain tumors. And I'll walk you through a little story of using CBTN resources for one such project. So one of my colleagues was studying neuroblastoma and had actually been working with some bioinformaticians from the CBTN and developed a pipeline to look for cell surface proteins that would be good targets to go after. And using that pipeline found glypican-2 or GPC2 as a good target in neuroblastoma. And he published on that and created an antibody drug conjugate with good success. And when looking at his paper, it spurred a thought in my mind of, well, this seems like a really great target to go after. I wonder if it's on pediatric brain tumors. So went to the CBTN. We could go to the next slide. Again, because the data is readily available, was able to very quickly query. So this is RNA sequencing data looking at GPC2 expression. And you can see that there's high expression, especially if you use 10 as a cutoff, that's what's being shown in B. You can see very high expression across the majority of embryonal tumors, ETMRs, medulloblastoma, and a subset of high-grade gliomas and diffuse midline gliomas as well. So this really piqued my curiosity and wanted to go after this target. Can go to the next slide. But what was really neat about the CBTN data was that you can dive as deep as you want to dive. And so we could look not only across different histologies, but actually within different subgroups. And so that's what's on the left. You can see the medulloblastoma, different subgroups, high-grade glioma subgroups, and embryonal type subgroups, as well as there were multiple cases that had both initial tumor as well as recurrence with RNA sequencing data. And you can see we could compare from the initial presentation to recurrence and seeing how GPC2 expression changed across time. Go to the next slide. Next, because again, the CBTN has so many wonderful cell lines available, we were able to get multiple cell lines, again here focusing largely on medulloblastoma and high-grade glioma, and could do flow cytometry to confirm that the expression was actually there, which we did, and again also using Western blots there in the bottom corner. Next slide. Oops, and then because I'm focused on CAR T cells, I was intrigued to go after this target with a CAR, and so developed 4 different CARs and focused in on 2 of them, which I have here in the red box. For the sake of time, I won't go through this in too much detail, but happy to talk offline about it later. You can go to the next slide. And again, using the cell lines that were available through the CBTN, we could do in vitro killing assays to see if these CAR T cells that we developed work. And they did. They worked beautifully across medullos, high-grade gliomas, and DMG cell lines. We can go to the next slide. And because we also have animal models available, we're able to do this as well in multiple. We have 2 medulloblastoma, that's what A and B are. And then the last one is a diffuse midline glioma, and we're able to actually show good efficacy in an animal model. And so this target, these CAR T cells have moved on. They have been licensed to a pharmaceutical company and are actively being pushed forward into the clinical trial space. And so again, because the CBTN resources were available, this was made much, much faster. So that way we could not only query this target, but get it tested, push it through in animal models, and then eventually out, hopefully here in the clinic in the very near future.
Great. What a whirlwind. But thank you so much, Dr. Foster, for sharing that really exciting story, and we're looking forward to seeing the publication once it comes out. But it's great to hear. Thanks so much for sharing. All right, so I want to move into our second chapter here of our presentation. So we offer these biospecimen resources. And so you might imagine, oh, I could collect this biospecimen and then I could do sequencing on it and then I could look at the genome of it. And so the great answer to that is that, well, we might already have that genome available. And so, you don't have to collect the specimen, you don't have to get the tissue, you don't have to, send it off to the sequencing center. We've already done that, and we're ready to share and work with everybody to be able to support your research even more rapidly. And so I'm hoping that one of my co-hosts here can respond to or share our next survey question about data. And so when I talk about data, I think lots of people might think of, you could have the command line, you have the little flashing dot there at the bottom, and these graphics of these laser light shows going back and forth. And so I want to emphasize that CBTN's data sets are accessible to everyone. And so if you are this, very incredibly competent bioinformatician who works at the command line and slings these workflows back and forth to analyze hundreds of genomes, we have the data and we have the resources to support you. But if you are not comfortable doing that, if you're more comfortable working in the laboratory and then you only log on to the computer just to check 1 or 2 things to support your benchtop research, we can support you too. And so we really support the full set of researchers, no matter where you are in that kind of spectrum of research, whether you're fully at the computer, fully at the lab bench, or anywhere in between. So our sequencing data overview. So what types of data do we provide? We provide all kinds of data. So we provide, of course, our whole genomes, as well as other different types that I'm going to get into here on the next couple slides. So what do we provide? So we provide genomic data, and so we provide that in alignment. So if you want to start with at the very beginning and do your own analysis, you can start from the alignment in BAM or CRAM format. And we provide both somatic alignments, so that's the alignment of the tumor tissue itself, or the germline as well. So that's the germline tissue in the participant, tumor and normal. We've already taken that alignment and we've run it through our pipeline in order to be able to identify variants, so points in the genome that are different from the human reference genome. And so we've already begun that first step, and that's available in GBCF format for our germline variants. And then we also have a very robust somatic variant pipeline, which runs through, I think, 8 or 9 different callers to identify both single nucleotide variants, copy number variants, and somatic variants. So we have a rich data set of genomic data available. If you're interested in gene expression, we have RNA sequencing data available. That's all the way available from the very, very beginning. You can start with unaligned reads in FASTQ format, or you can pick it up at different steps of our RNA-seq workflow, either in aligned reads format or in our process data, where we've already counted and looked at the different genes and saying, which ones are expressed highly, which ones are more lowly expressed, how is expression changing in these different tumor types, and how are these different genes might be fused together, which could implicate perhaps a drug target. So these are, I would say, are our two main data sets, but we also offer some other data sets as well. We offer a collection of proteomic data, which is available in a couple different formats as part of our analysis pipeline. We also offer imaging data, and so this is a new data type that is growing more and more. So we have radiology data. So if a radiologist has taken MRI scans of a patient, we have those images, and then we also have the report that the attending radiologist wrote up, from reviewing those images themselves. So that's paired 1-in-1, so you can look at the image and read the report at the same time. In a similar vein, we have histology images as well. So if a tumor has been put out on a slide and stained, and the histologist has reviewed it, or pathologist has reviewed it, you can look at those same stained slides, and you can read the report by the pathologist, to compare back and forth and make those same observations. We also have surgery and operations reports, and so from that surgeon who was working, who operated and performed the procedure on the patient, we have their write-up, and their report is sometimes available as well. And then finally, we have other data types. So we do have some microRNA sequencing, and we have some single cell sequencing for some participants, but this is not necessarily available for all participants. And so that's what we have available for some other data types. Okay, so this is all of our data. And so where does this live? So how can I access this robust collection of resources here? So CBTN manages 4 what we call platforms. So this is part of our data infrastructure. And so we have 4 different websites, 4 different platforms that we use, and each one has its own kind of strengths that facilitates different research in different ways for users to be able to carry out their research. And so the first one is the Kids First Data Resource Portal. And so this is a tool that allows users to be able to identify different biospecimens and different participants for their research. And so here, for example, here's a snapshot of the data resource portal. It's very visual, it's very colorful, and that once you're looking in the CBTN study, you can then add different filters. So for example, if I research ependymoma, you can just click this one bar and you're going to look at just the ependymoma cases. There's also clinical filters up at the top, or demographic filters if you want to narrow down your search. So it allows you to be able to identify the participants that have enrolled in CBTN in order to be able to identify a subset that might be most relevant to your research. So once you identify those participants, then you can take that set of participants and basically say, show me the files, show me all the different data files that we have that are available for this set of participants. And so that takes you over here into the file repository, where you can see, individual files listed here. So here are different variant calls. This is an expression file. This is from an RNA-seq analysis. And then you can download those files and begin your analysis. So it takes you from the viewing as the participants to now you're viewing the files. And so emphasize up here at the top, access this today. So the Kids First Data Resource Portal is open access to browse, and a lot of our files are open access as well to download. So you don't need to even apply to be able to get into the Kids First Data Resource Portal and begin exploring and seeing what does CBTN have to offer. Our second tool that I want to emphasize is one that I really love. It's called PEDcBIOPORTAL. And so if you're familiar with the website cBioPortal, which is a tool for visualizing cancer data that's been published in publications, PEDcBIOPORTAL is the same tool, but what we've done is we've loaded the CBTN data into it. And so you're able to view and browse variants within your different CBTN samples. You can also begin to compare different outcomes within participants as well. So when you pull up the CBTN data set, which is part of the Pediatric Brain Tumor Atlas, it begins with this very large set. So it describes, the participants, all of the patients that we have genomic data for, and then it gives you information about all of the different mutations that might be present in that full data set. And similar to Kids First, you can narrow this down more specifically. So if you're just interested in medulloblastoma, you can apply a filter and just look at that. But once you want to learn more, if you study a gene of interest, so for example, here's TP53, you can look at the mutations within that particular gene. Again, it's very visual. So we have the different domains of the protein labeled here. And then each of these markers corresponds to a known mutation that we've observed in the CBTN cohort. And then it's annotated as well. So not just that these mutations are present, but telling us that, yes, this is the very specific protein coding change that we've observed. And then some annotation information as far as what does this mutation mean as far as the possible cancer causing state of this particular mutation. But more than just looking, you can do analysis in PEDcBIOPORTAL as well. And so you can compare different data sets. And so by looking at, across different tumor types here that I have on the bottom, I'm looking at a number of mutations across these different tumor types. And so you can do comparison, you can form groups of patients, you can find patients that have the mutation but don't have a mutation and see how do those groups compare. And so it allows you to do this analysis in a point and click all in your browser manner. And then finally, the last view I'll show you from PEDC is that it allows you to be able to view what's called patient view here. And so here we're looking at a single participant. And then for this participant, what we have outlined is this is all of the mutations that we've observed as part of our CBTN data processing workflow. So we've already done these first steps of identifying, what are the mutations that this patient and this sample have in their genomes. And so it allows you to be able to, find a mutation very, very rapidly. And so if you study a gene and you have a particular patient who maybe has a mutation in that gene, and you want to compare it to our CBTN data set and say, have other patients, are there any other patients out there that have had the same mutation? How were those patients treated? What were their outcomes? We can support that kind of research by looking in our data sets here and comparing what you're bringing to us to what we have in CBTN. The third tool I want to talk about is CAVATICA. So CAVATICA is our very large-scale bioinformatic platform. And so it allows you to be able to run these big bioinformatic workflows in the cloud. And so it allows you to be able to bring in these. So I think here we've got it looks like an RNA-seq workflow where we are running STAR, and then we're going to pass that on to STAR Fusion. So it allows you to be able to run these big software tools to do these kind of big data alignments and variant calling and that sort of thing that you might be doing already in the cloud or perhaps on a local high performance computing cluster at your own institution. And so you can run these big analyses on CBTN data, or you can bring your own data as well into CAVATICA. But what's great is that also in the cloud, it allows you to be able to analyze that data as well. And so you can work either in RStudio or Python notebooks. So once you generate that output, you still need to make the graphs, right? You still need to look more carefully at those variants. And so you can do all of that in CAVATICA without having to review or download to your local computer in order to be able to do that type of data processing. So you can do it all in one space in the browser as well. And then the final platform I'll talk about is Flywheel. So Flywheel is our newest platform that we've just gotten started. Flywheel is our imaging platform. So if you were, you perked up when I was talking about those radiology images, then this is where we're going to help you get set up in the future. So Flywheel allows you to be able to view all of this imaging data within your browser, and it allows you to build similar analysis workflows for your imaging projects. And so, you can click in and then all within your browser without having to download any files or move anything, and then open it on your computer. You click one button, on here, and then it takes you over, and you're bam, you're looking at it. So the data is all very accessible and very easy to scroll through as well. So hopefully from that sense of overview there, we've got lots of different data available, and we have lots of different ways that we can share that data with you all. So again, wherever you come in on that kind of data use spectrum that we like to talk about and think about, we can support your work as early career researchers. And so I want to pass the mic now to Adam Walker. So Adam has been a CBTN user for a long time, and he's attended a lot of our office hour sessions. So I asked him to join us today and tell us a little bit about his research and how he uses CBTN data as part of his work.
Does the video work? Oh, okay, perfect.
Hello.
It does. Hello. Hi, everyone. I'm Adam Walker. I am a research assistant within the Department of Pathology and Lab Medicine at the Children's Hospital of Los Angeles. And as David just mentioned, I have been using a lot of these online portals to assist with some of my own research projects, as well as other research projects within our department more broadly. So my specific focus happens to be on digital pathology and imaging pipelines. And we have been able to utilize CBTN a lot for that, as well as utilizing the data set for a broader range of projects, as well as some literature review projects. And that's where I'll start right now. Oh, I can even change the slides. So our first focus here, one of the researchers, Dr. Ellen Chang, within our department, was studying IDH mutant brainstem gliomas. And the more that you add onto the beginning of gliomas, the more specific you get. The IDH mutant brainstem gliomas within children, these are very, very rare tumors in a very rare part of the brain. And as such, they're very hard to find. And so we did a literature review of our samples here at Children's, and we came up with 3 good samples in the past few years. But as a part of this project, we really wanted to do a comprehensive literature review and see what other institutions had in their biobanks as well, and see if their data and their samples corroborated with what we were finding at our institution. And so we were able to utilize the Kids First data set and go in through the PEDcBIOPORTAL and be very specific in what we were looking for, and that we wanted IDH mutant brainstem gliomas with a very specific mutation, and we were able to narrow it down to just a few samples and add those to our project as a part of the literature review and corroborate with what we were finding, which was incredibly valuable in backing up all of the data that we had. Our next project that we want to focus on here is more pertaining to me and more speaks to really how multimodal the CBTN data set is. As David was just going over, there's many different ways of attacking all of the data that is there through many different portals, as there's just a lot of data that you can work with. So my project specifically deals with histology, and within the CBTN data set, pretty much every single one of these patients has a corresponding H&E slide with whatever tumor sample they are, whatever tumor sample has been submitted. So here we have an example of a H&E of a pineoblastoma, which is another rare tumor and another very specific part of the brain. But the CBTN data set allowed us to get a larger cohort that we can use as a part of some of our machine learning models, which again, we can add, in this case, around 700 patients to this data set so that we can better train our algorithms or better use them as tests for our algorithms to make sure that we really have the robustness needed in all of the analysis that we're doing. And for this specific project, we're looking at tying together those histology images along with the specific genetics of these tumors. And so the CBTN data set, again, has all of this multimodal data that we can go in and really have this paired H&E slide image. In this case, they're .svs files. If anyone is familiar with digital pathology, it's a burgeoning field, I would recommend. And these genetics, wherein there are all the way up at BAM files, which are very, very large files, jam-packed with all sorts of genetic information that can then be further analyzed on platforms like CAVATICA, which we have used at our institution, as well as more already processed samples like VCF files, which have already been run through a lot of the processing algorithms by the CHOP team. And they're a lot smaller file sizes such that you know that they're already analyzed and you can more easily move them around. So those are just two of the main projects that we have going on so far at Children's, but we have probably over a dozen projects that have either been completed in the past couple of years or are actively in process, where we're using the CBTN data as a backbone resource for a lot of the research that we're doing as a means of doing literature review and really backing up what we're finding or adding robustness to any of our projects. So how has CBTN really helped us along the way? As David stated at the beginning here, I've attended a lot, and I mean a lot, of the office hours, and the team could not be more gracious and helpful along the way at every single one of them. I think I'm there pretty much every month, just working through exactly new projects that have sprung up as I quickly became the point person at Children's within anatomic pathology. A lot of people come to me for help with navigating this data set and helping them get exactly the cohorts that they need. And over here on the right, fast email correspondence. I can assure you these guys get back to you in a day, 2 days at most, which has just been really nice in helping out with all the projects that we're managing. And that even goes into project-specific Zoom calls. If something doesn't work out in one of the office hours or an email correspondence, they're more than happy to jump on a private Zoom call. And they have in the past a few times with helping out with getting our stuff along the way. So really, it has been a fantastic time working with this data set. And I'm sure, I think we're answering questions later on, so.
Sure. Awesome, thanks so much. Yeah, so let me wrap us up here and get us towards the end. So you might be wondering, okay, so how do I access details? And so I've seen some questions in the chat that have already come up. And so yes, you can access this data. So you do not need to be, that we showed that globe at the beginning with the pins of the different sites. You don't have to work at one of those sites to be able to apply and benefit from CBTN data. So we empower researchers around the world. If you're out there and you have this share our mission to try to help kids, then we want to help support your research. And so all of this is easy to use. Hopefully it's all open access. It's all free. We're not looking for anything in return. It's your research, it's your publication. We just want you to maybe acknowledge us in the acknowledgement statement. That's all that we ask in return for this great data set. Okay, so how do I access this? So if you're interested in accessing samples, you can browse and search different clinical and phenotype data in the Kids First portal to kind of get you started and see what's available. And then we have a REDCap form / CBTN request that's also on our website, cbtn.org. That comes to me and a whole handful of folks here at CBTN who review it in order to help you identify that cohort and kind of confirm it and say, yep, these samples look great for your research project. We pass it up to our scientific committee, and so they review it. So this review is meant to be, it's meant to be constructive. It's meant to be, you're getting senior researchers looking at your data, and it's all meant to be, or not at your data, at your proposal, and it's meant to be a very positive, supportive experience where we, help refine and say, oh, did you think about this too? Here's these samples as well. And so it's meant to be a really positive experience that usually returns a decision in 4 to 5 weeks. So our goal that we work towards is that once approved, we hope that samples will be shipped within 6 to 8 weeks from the time that you hit the submit button on our REDCap link, pending making sure all the agreements are in place between the different sites and institutions. The data is even quicker, and so as I emphasized earlier, we have a lot of open access data that's already available. So you can go and log in right after this call and start looking at those different variants that are already available. So those somatic variants, all types are available, as well as our processed RNA-seq data. You can access this on the Kids First portal. You can also access that visualized format with the with the genes spelled out and the different markers and things like that. That's available on PEDcBIOPORTAL as well. So those are two sites where you can already access this open access data. If you want the controlled access data, so this includes things like, if you want to start with the alignments themselves, or you want to access the germline variants, those are available through application, and it's the same application link, CBTN request, that's also available on our webpage. Once you submit that assigned data use agreement, we look to review it in about a week and then respond and pass it up for legal review and authorization within about a month total. And so if you have any questions or need assistance at any step of the process, from how do I start to where is my application, you can always reach out to us at research@cbtn.org. And so that inbox goes to me and a handful of other great folks who are all gonna help review it and review your request. Okay, so then in my final couple of minutes here, I'll just share some last minute things about how else do we support investigators and collaboration opportunities. So I'll give one last plug for the CBTN office hours session. And so each month we have an office hour session. It's usually a smaller session where we just go around the room and talk to everybody who's there. And if anybody has any questions, then we help them get started. So the questions are, we have a whole panel of folks. So we have folks who help people review their applications and submit. And that stage, we have members of our data team who are available to answer questions about the data. We have members of our bioinformatics team who developed the pipelines and generated all these variants. So no matter what your question is that you're bringing to us, we're able to answer them at the office hour sessions. So these are always held the fourth Tuesday of every month. And so we'll have the next one at the end of October. Details for this are available on our social media accounts, or you can ask to be invited to a recurring calendar invite, and we'll make sure it shows up to your inbox. Again, research@cbtn.org, and we'll help you out. CBTN also helps facilitate PNOC, what we call the PNOC working groups. This is part of the Pacific Pediatric Neuro-Oncology Consortium. And so we have working groups that are coordinated mostly based on different histologies. So there's a ependymoma working group, there's a medulloblastoma working group, and so different working groups based on the different histologies. If you want to be in the room with different researchers who are studying that same disease that you study and grow your network and meet other folks, and this is a great way to get involved in that. So we help organize these working groups as well. We also support Open PBTA. So Open PBTA is a community of researchers that are actively researching CBTN data. So it's this open collaboration. We have a community of folks. They've already begun processing the data. So if you need, even further downstream analysis, so if you're interested in expression analysis, tumor mutational burden, immune deconvolution, or molecular subtypes about our different CBTN data sets, we've already produced that. And so you can meet up with these wonderful folks over at Open PBTA. They have a GitHub space under Alex's Lemonade Stand, which is where they like to collaborate and talk to each other as well. But we help support them and their crowdsourced effort towards working towards these solutions for pediatric brain cancer. And of course, all of our folks that we have in our CBTN community. And so, of course, CBTN is all about this sense of open access and collaborative spirit. So we realize that the mission that we're looking to find is that, an end of pediatric cancer for all children everywhere is of course something it's too great for any one institution to be able to solve on their own. So that's why we need our community partners. We need our member institutions. We need our new investigators that are just getting started. We need everybody to be able to work towards this great mission that we have. And if you want to hear more, then you can come if you're attending ASHG, the ASHG conference. We're going to be giving a presentation specifically about the Kids First Data Resource Portal. And so that'll be at 2:30 on Tuesday, October 19th. And so if you're attending ASHG, the virtual meeting this year, then we can come say hi to our collaborators with the Kids First program, and they can talk a little bit more about those resources and how they connect with CBTN's resources. All right, so we've got a couple minutes left here. And so I know that we've got some questions in the chat, but happy to answer any other questions that might have come up since we've gotten started. I'll just review and make sure we've got all the questions that we've answered so far. So I see that Nicole asked about, hi Nicole, you asked about glioma data that's annotated with the region. Yes, and so all of our samples are annotated with a field that indicates what part of the brain the tumor was where the tumor arose. And so that information is collected and it's associated with our different biospecimens in our data set. So you will be able to review that as you're reviewing our different CBTN specimens. Then Andrea asked, does our institution need to be among that initial list, or can anyone join? No, it does not. Anyone can request. And so anybody can submit an application to receive and use CBTN data. Again, we want to welcome everybody. This is a big problem that we're all trying to solve. And so anybody can come. Please let us help you. Tell us how we can help you best. Anybody can come. Robert is asking who could be a contact person regarding initiating discussions about becoming a member site. Yeah, and Jerry and Kamna responded, email us at research@cbtn.org. So that's a great way to communicate with us in order to be able to, any questions that you have, research@cbtn.org is a great place to send them. And so we've got about a dozen folks who all have access to that email address. And then from there, we can, read each one and send them out to the correct team of CBTN folks to be able to respond. How does CBTN make sure that samples are not used for redundant experiments? Yeah, that's a great question. So we actually collect a manifest. And so it's part of our clinical data is that every time we have that, send out that sample, we make a note of it. And so each of our CBTN projects that we support is a list of projects. And then we, every time we send out that particular sample, we write down that project ID. And so we're able to review that. And then as we're reviewing it, if we find that a proposal comes in about a similar, with a similar research asking for similar experimental data, we might try to connect those two researchers and say, oh, this researcher, you're doing the same type of thing as this person. That might be a great collaboration in the future. So we do monitor that as part of our analysis. Are there any plans to do RNA-seq on matched normal brain samples? Yeah, Robert. So not at this time. So right now, the RNA-seq that we have is just available from the tumor itself. So we don't have any matched normal brain samples. We might point you towards, I know there are, normal repositories out there like the GTEx repository. And so we can work and collaborate that set of resources. viability frozen cells of tumor tissue available to share? We do offer cell lines. So we have cell lines that are, cultures that are still growing, and so we can subculture it and then submit that as if you want to then grow up that cell culture as well. And then we also have flash frozen tissue. Thanks, Kama, for posting that. So we do have a lot of different resources. Okay, I think I got to everyone, and I'm just about out of time here. So maybe I will pass it to Jenna if you wanted to wrap us up here with a little bit, some final closing thoughts about CBTN.
David, I think that Jenna might have had to jump, so you can.
Oh, I'm sorry. Yeah,
no, that's totally okay. Thank you.
Great. Yeah. So thanks so much for joining, and thank you, Erin. So Erin just posted the last poll, and so if you have any, this is basically just how can we help you even further. And so if you just want, kind of an email write-up, we can send that. If you want to have a one-on-one call with us, we'd love to, talk with you further. If you want to just come next month to our fourth Tuesday office hours, we can set up with you there. Lots of different ways that we can connect with you further. But I want to end with our promise. And so we know, especially for early career folks, that there's so many demands on you as far as getting your research up, finding these discoveries, writing these publications, getting these grants. There's a lot. You're being asked to do a lot. And so we want to help you. And so we want to provide you fast and free access to these data and specimens. We want to support your grant writing, your grant opportunities, whether that's a federal grant, like an R03 grant, or a philanthropic grant, or your research, at your institution. And so all that we ask back from you is that, whatever you discover and whatever data you generate, you share any data back with us, and then you share and publish with the whole world. So that way everybody can learn, about the discoveries that you're making for this, hugely important mission of ours. And so together we're going to collaborate with researchers of all levels around the world, and then we want to provide better treatments to children. So thank you so much for joining us this afternoon. I think I'll wrap it up there. We'd love to talk with you further. So please connect with us either by emailing research or connecting at one of our events in the future. But overall, thank you again for joining this afternoon, and I hope you all have a great day.